Death is not a moment. That's the thing almost everyone gets wrong. We imagine it as a line, one side alive, the other side not, a switch, a threshold. But biologically, physically, at the level of cells and chemistry and the atoms that make you up, death is a process, a cascade, a slow unwinding that begins before the last heartbeat and continues for days, weeks, years after. And when you follow it carefully, all the way down, it stops being a story about ending and starts being a story about what you actually are. I'm Paul Dirac. Let's think about this without flinching. The heart stops, or breathing stops. Usually one triggers the other within seconds. The delivery of oxygen to the body's cells is interrupted. And here's what happens, not symbolically, but mechanically, in the minutes that follow. The cells don't know yet. That's the strange part. For the first several minutes after circulation stops, cells continue attempting their normal operations. They're still burning ATP, adenosine triphosphate, the chemical energy currency of every living cell. They're still trying to maintain the ion gradients across their membranes that allow them to function. They're still signaling, still processing, still behaving as though the system they're part of hasn't just lost its central coordination. The brain, which consumes more oxygen per gram than almost any other tissue, starts failing first. Neurons begin losing the ability to maintain their electrical charge within 20 to 40 seconds of oxygen loss. Consciousness fades. But here's what matters. At this stage, the process is partially reversible. People have been resuscitated after 4, 5, even 10 minutes of cardiac arrest and returned with intact cognitive function. The brain was failing. It had not yet failed irreversibly. Death at this stage is not a fact. It's a direction, a trajectory. One that can, under the right circumstances, cold water, rapid intervention, targeted resuscitation, be redirected. The boundary between life and death at the cellular level is not a wall, it's a gradient. But the window closes. And here's what happens when it does. Without oxygen, cells can't run oxidative phosphorylation, the efficient pathway for making ATP. They fall back on anaerobic glycolysis, a backup system far less efficient that generates lactic acid as a byproduct. It buys time. A few minutes in most cells, a little longer in others. But eventually ATP runs out entirely. And when ATP runs out, the pumps stop. Your cell membranes are studded with protein pumps that actively maintain the electrochemical environment inside the cell, keeping sodium out, keeping potassium in, keeping calcium at extraordinarily low concentrations. These gradients are not passive. They require constant energy expenditure to maintain. The moment ATP is exhausted, the pumps fail. Sodium floods in, water follows the osmotic gradient, rushing into the cell. The cell swells. Calcium, normally present inside cells at concentrations roughly 10,000 times lower than outside, pours in uncontrolled. Calcium, at those concentrations, is a catastrophe. It activates enzymes called proteases and lipases, enzymes that break down proteins and fats. Under normal circumstances, these enzymes are tightly regulated. Now, with calcium flooding the cellular interior, they activate indiscriminately. The cell begins digesting itself, using its own machinery to dismantle its own structures. This process is called autolysis, self-digestion, and it is not external attack. It is the cell's own chemistry, untethered, running without regulation. The body is not being destroyed from outside, it's being dismantled from within by its own tools. A few hours later, something visible happens. The muscles stiffen. Rigor mortis. Most people know the term. Almost nobody knows the mechanism, and the mechanism is the interesting part. Muscle contraction works through interactions between two proteins, actin and myosin. Myosin heads attach to actin filaments and pull, generating force. This requires ATP to form the cross bridge, and it requires ATP to break the cross bridge and release. Both the formation and the release of the bond are energy-dependent processes. Without ATP, myosin heads attach to actin, because the calcium flooding from autolysis triggers the contraction machinery and cannot let go. Every muscle in the body locks simultaneously in contraction. The jaw, the hands, the chest, the legs, all of it. Rigor mortis is not death freezing the body in place. It's the contraction cycle running with no release mechanism. The machinery only knows one direction. It reaches peak stiffness at around 12 hours, then gradually resolves over the following 24 to 48 hours, not because anything is recovering, but because the proteins themselves are beginning to break down, the actin and myosin degrading past the point of maintaining their locked configuration. Even the stiffness of death is a chemical process. Everything is. Now, here's the section that I think changes how you understand what you were while you were alive. Your gut contains somewhere between 38 and 100 trillion bacteria. Your microbiome, the community of microorganisms living in and on your body, is, by cell count, roughly equal to the number of human cells in your body. These organisms coexisted with you. You provided them an environment. They assisted your digestion, trained your immune system, produced essential vitamins. A genuine ecological partnership maintained over your entire lifetime. While you were alive, your immune system maintained the boundaries. Intestinal walls kept gut bacteria where they belonged. Immune surveillance prevented colonization of tissues that were supposed to remain sterile. The ecology was managed. When you die, management stops. The intestinal walls, no longer actively maintained, begin to fail. The bacteria migrate. They spread through the intestinal wall into the surrounding tissue, into the bloodstream, into the organs. They begin consuming the nutrients that autolysis has released. The decomposition of the body from within is, substantially, the microbiome that lived inside you being released from the constraints your living body imposed. You don't decay because something foreign invades you. You decay because the internal ecosystem you maintain throughout your life is finally, without your active management, allowed to follow its own logic. And what follows, what we call decomposition, is not destruction. That framing is wrong, and it matters. Every atom in your body is going somewhere specific. The carbon in your muscle fibers will be metabolized by bacteria, released as carbon dioxide, absorbed by plants, incorporated into sugars, eaten by animals. The nitrogen in your proteins will cycle through soil organisms into the nitrogen pool that living systems draw from. The calcium in your bones will dissolve slowly over years or decades and re-enter geological and biological cycles. The phosphorus that carried your genetic information will become available to new organisms building their own. Nothing is lost. Nothing disappears. The atoms you borrowed from food, from air, from water over the course of your life are being returned to the systems they came from. Decomposition is matter finding its next assignment. It is the most efficient recycling process on Earth, refined over billions of years of evolution, involving bacteria, fungi, insects, scavengers, an entire decomposer ecosystem whose existence depends on and enables the continuous cycling of matter through living systems. You did not own those atoms. You organized them, temporarily, into a pattern. Death is when the organization releases its hold and the atoms resume their older, broader circulation. Now, the brain. The thing that more than any other organ we associate with what makes you you. The brain is the first organ to show functional failure after oxygen loss, and in terms of the specific injury that makes death irreversible, it's the critical organ. Clinical brain death, the legal and medical standard used in most countries, means the irreversible cessation of all brain function, including the brainstem. No electrical activity, no reflexes mediated by the brain, no response to stimulation. But brain death as a medical declaration is not the same as the immediate cessation of all neural activity. After cardiac arrest, and even after clinical brain death, individual neurons can survive for hours. Spinal cord reflexes, mediated at the level of the spinal cord, not the brain, can persist for some time. Glial cells, the supporting tissue of the brain, survive longer still. And here's the genuinely uncertain part, where I want to be careful to distinguish what we know from what we don't. There is a small body of research, including a 2023 study in PNAS, documenting surges of high-frequency, coordinated electrical activity in the brains of dying patients in the minutes immediately following cardiac arrest. These surges occur in regions of the cortex associated with conscious processing. What they mean for subjective experience, whether anything is experienced, is unknown. The researchers who documented these patterns are careful not to overclaim. The honest position is, we don't know. We can measure electrical activity, we cannot measure experience. And the relationship between the two is one of the deepest unsolved problems in all of science. What happens inside the brain at death, whether the lights go out all at once or whether there's a final flare, we genuinely don't know. Now let's pull back to physics, because physics has something precise to say and something honest to admit. The atoms that composed your body continue. Conservation of mass and energy, they don't cease to exist. The matter is not annihilated. The energy stored in chemical bonds is released and redistributed. By every conservation law we know, nothing that was in you is gone from the universe. It has changed form, changed location, changed organization. But the pattern, the specific, extraordinarily improbable configuration of roughly 37 trillion cells, 86 billion neurons, their specific connectivity, the synaptic weights encoding your memories, your personality, your particular way of processing the world, that pattern disperses. The information that constituted you becomes distributed across an increasingly large number of molecules in an increasingly large number of locations. It isn't destroyed in the sense of violating any physical law, but it diffuses, it entangles with everything else. Recovering it, reconstructing the pattern from the dispersed atoms, would require information about the position and state of an astronomical number of particles. It would be, in any practical sense, impossible. In any physical sense, irreversible. The pattern ends. And the pattern was, at minimum, where you lived. Here's what I want you to sit with at the end. You are already, right now, in a continuous process of dying and renewal. Every day, roughly 300 billion of your cells die and are replaced. The cells lining your gut replace themselves completely every few days. Your red blood cells live about 120 days. Even your neurons, long considered permanent, show some degree of renewal in certain brain regions. The atoms in your body turn over continuously. The you of a decade ago shares almost no physical matter with the you of today. What you are is not a fixed collection of matter. What you are is a pattern, maintained actively against entropy through a continuous process of replacement and repair. The thing we call death is just the point where that maintenance process stops, where the energy input ceases, where the pattern can no longer sustain itself against the thermodynamic tendency toward disorder. And the pattern, without maintenance, disperses. So here's the question that death actually opens up. It's not what happens to your body when you die. We've answered that in outline. The real question, the one underneath it, is what exactly was alive? What is the pattern that dies? Where does a pattern end and the substrate begin? Is the pattern you, or is it something the atoms were doing temporarily? Physics can describe the cascade. Chemistry can map the autolysis. Biology can trace the microbial succession and the nutrient cycling. But none of them can tell you what the pattern was in the sense that would tell you what is lost when it goes. That question, what you are, is the one that death makes urgent. And honest science, as of today, does not have a complete answer. I find it remarkable that the question, what happens when you die, leads here, not to comfort, not to terror, to a genuine, precise, unresolved mystery about the nature of organized matter, information, and what it means for a pattern to be alive. The atoms continue, that much is certain. What continues with them, if anything, remains the most important open question there is.